Okay, today we're going to talk about a second group of organic molecules. We talked about carbohydrates already, and this group includes oils, butters, lard. You can probably guess this is fats, lipids, oils, steroids. The whole group is actually called lipids, which includes fats, oils, and steroids. And the defining characteristic of all lipids is that they are nonpolar. So if you look at this fat molecule here, you see a lot of carbon, hydrogen, carbon, hydrogen. You see a couple oxygens in there. But do you see this hydroxyl group? Remember, the hydroxyl group on carbohydrates makes it polar. It's a polar group. No, you don't see any of them, right? So no hydroxyl groups, primarily non-polar bonds between carbons and hydrogens. Important thing about them, though, the carbon-hydrogen bond does have a lot of energy in it. So fats and oils are really important because of their high energy value, twice the amount of energy as carbohydrates. And that's important. So we tend to see them as long-term energy storage, especially in animals. Okay, so the two main types of, or the basic unit that we see in fats and oils are fatty acids. One of them is called the saturated fatty acid, and the other is called an unsaturated fatty acid. And we put them into these polymers. In this case, the polymer is only 3 fatty acids connected to a glycerol, and it's called a triglyceride. So the basic fat molecule is 3 monomers or 3 fatty acids attached to a glycerol backbone, and we call that a triglyceride. When we looked at the fatty acids here, just to go back, you notice the hydroxyl groups on there. So you'd think, well, aren't they polar? Shouldn't they be polar? But remember, those hydroxyl groups get removed during this dehydration synthesis, or this condensation, pulling out water to covalently bond these monomers into a polymer. So that's why we don't see that polar group on the fat molecule. Okay. And again, 2 types of fats, saturated and unsaturated. You probably recognize oils as unsaturated. In fact, canola oil is a polyunsaturated fat. Saturated fats tend to be things like butter, lard, animal fats. They're not as healthy. They clog our arteries. They create much more problems health-wise. It's the unsaturated fats that are the healthier ones. And when you look at food labels, we actually list the total amount of fat and the amount of saturated fat. And if it's not saturated fat, it's hopefully unsaturated fat, although there's a third type of fat called a trans fat, which we'll talk about in a little bit. So again, when we look at the structure of saturated fats versus unsaturated fats, saturated fats are very linear. They're straight, just like this diagram shows, and they tend to pile together and to be very kind of tightly packed together. And there's not a lot of room between these fatty acids. So they tend to be very dense, and we see them as solids. Unsaturated fats are kinky. They have lots of kinks and bends in them. And these kinks mean that they don't fit together very well. And because of that, there's more movement amongst these fatty acid chains, and they're liquids. So when we see these diagrams, when we see that the unsaturated is bent, they're actually bent and kinky like that, and they don't pack together well. That's why they have more movement in the fatty acids, and they remain liquids at room temperature, whereas butter, lard, and other saturated fats remain solid. There's another type of unsaturated fat called a monounsaturated fat. That's one double bond. That's an example. An example would be olive oil. Olive oil, if you put it in the refrigerator, it freezes, right? So it's closer to a saturated fat because it just has one double bond, but it's still, that one double bond makes it a liquid at room temperature. So we have monounsaturated fats like olive oils, and then polyunsaturated fats, many double bonds. Most of the vegetable oils are polyunsaturated, and they will remain a liquid even in the refrigerator because of the kinks and all the double bonds in there give them a lot more room to move. Okay, so saturated fats, unfortunately, are something that our body craves. They used to cook McDonald's French fries in lard. People loved it. They switched to oil, and they just tasted greasy, and you had more grease on your fingers, and everyone said that French fries cooked in oil and polyunsaturated fats, though it's healthier, they just didn't taste as good. So chemists got busy and they said, well, we can take polyunsaturated fats and we can hydrogenate them. We can break these double bonds and we can make them more like saturated fats. And then when they used these hydrogenated oils, they tasted more like lard. And we still have like peanut butter is hydrogenated, so we don't have the peanut oil floating on the top. It's all creamy smooth like butter. Unfortunately, when you create that transfiguration, you can see saturated fats, the hydrogens are on the same side of the double bond. Trans means across. So when we hydrogenate these polyunsaturated fats, we get these trans fats. They're very unhealthy, and you can see this is a joke about New York City. A year or two ago banned trans fats in restaurants. So you can't, if you go to a restaurant in New York City, they're not allowed to use any trans fats in their food. So we're kind of aware of trans fats. They're man-made, they're unhealthy, but they're made by taking a polyunsaturated fat and basically using heat and pressure to break those double bonds and put these hydrogens on to create that trans configuration. So where do you get trans fats in your diet? Well, it's all the food you love, right? It's baked products, it's some animal products, cakes, cookies, fried potatoes, anything cooked with, Crisco or anything of these sort of shortening type products, margarine and so forth, especially stick margarine because it's much more firm than tub margarine, has a lot more hydrogenated oils and trans fats in it. Anyway, stay away from the trans fats. Other important lipids, though, this one is two fatty acids attached to a phosphate group. Phosphate is a polar polyatomic ion, and it's attached to these non-polar fatty acids, and it's called a phospholipid. It's an important molecule in our cell membranes. If we have a non-polar region and a polar region, the polar region is going to be attracted to the water, the non-polar region is going to be attracted to other non-polar molecules like lipids, and it's going to form this lipid bilayer. So this is an example of the cell membrane composed of this lipid bilayer. And again, phospholipids are important because they make up the majority of our cell membranes. Here's an example of that phospholipid bilayer where the phosphate heads are on the inside and outside of the membrane. Again, our body's 70% water, so the area inside and outside cells is predominantly water and aqueous solution. So the phosphate heads are attached to that or attracted to that, and the lipid tails are hydrophobic, water hating. They're inside, away from the water. Last group of important lipids are steroids. Now, you've heard about steroids. You've heard about Lance Armstrong. He got all of his title stripped from the Tour de France. Barry Bonds. There's just a lot in the news. These are synthetic steroids, but real steroids are produced by humans and other animals, and they are important sex hormones. So here's an example of one of those sex hormones. This is called testosterone. This is the one that they've produced in labs, and a lot of athletes have been taking to get an unfair advantage. Higher levels of testosterone increase muscle mass and recovery from hard workouts and give athletes an unfair advantage. So estrogen, testosterone, you can see they are ringed lipids, so they don't have that fatty acid chain, but they do have carbon and hydrogen bound together like other lipids, and they are nonpolar. So though they have a very different function in terms of being hormones, they have, again, that nonpolar characteristic and composed primarily of carbon and hydrogen. And that's a quick review of the important lipids that we'll need to know this year in AP Biology.